I'd like to introduce our moderator for the rest of the morning. His name is Dr. Bryce Richardson. Bryce grew up here in southern Idaho. He holds a BS in biology from the College of Idaho in Caldwell, an MS in forest research from the University of Idaho, and a PhD in plant pathology from Washington State University. He is currently a research geneticist with the Forest Service Rocky Mountain Research Station in Provo, Utah. Bryce?
Good morning. Thanks, Tom. Let's see here. Okay, so I guess to start off with, I'm going to make the argument. Hold up. I'll make the argument that to really do restoration to be successful, we really need to understand what our species are, what the boundaries are between our species, and also where the populations exist within species. And this is both a morphological question, also a molecular question. So I'm not going to spend too much time up here telling you about sagebrush. You guys probably know it better than I do. Its importance to ecology. But I will just kind of review the taxonomy that we use to distinguish different subspecies of sagebrush, big sagebrush. Artemisia tridentata are basin subspecies. And it's pretty obvious when you see these plants. Usually they're quite tall, have upright branching. And you can also do some chemical tests. UV fluorescence is negative with this, so I just have an example down here at the bottom. And you can find both diploid and tetraploid plants that fit this morphology. Vaseina is shorter, flat-topped, and then it has this positive UV fluorescence, which is right down here at the bottom. It again can exist as diploid or tetraploids. And finally, probably most important to a lot of lands that we manage is wyomingensis. And it tends to have morphology like Vaseina. But oftentimes it has no or mixed UV fluorescence. And it's thought to be entirely tetraploid. And most of you have done this or come across examples where this isn't always straightforward. There's exceptions to these. And it's led some people to name other subspecies, which might be true. But I thought I'd present this picture to you. This is just a demonstration garden we have in Provo. We're taking one plant from each of our seed sources for a common garden and just planting them out according to subspecies. So we have Vaseina here on the left side and our tridentata subspecies on the right. And in the middle, we planted all our tetraploids. And you can, if you look down the rows, you can see the amount of variation that occurs just in these tetraploids. And toward the back here, you get ones that are almost undistinguishable to the diploid tridentata. So enormous amount of variation just in these plants. And of course, some of these you would consider to be wyomingensis, and some of them you'd consider to be tridentata. So why is polyploidy important in this species? So polyploidy or genome duplication can radically change the morphology and the tolerance to environmental stresses, principally by altering gene expression. And polyploids, in this case tetraploids, including wyomingensis, are probably more abundant on the landscape than diploids. And this might be the case just because most of the lands that would support diploid plants are now under the plow, are being used for agriculture. So when Grant MacArthur went out and surveyed thousands of plants, he found that over half of them in various locations were actually tetraploids. So we really don't have a grasp on what subspecies wyomingensis is. We know that this process of genome duplication only goes in one direction, that we start with diploid plants and end up with tetraploid plants. We don't find triploids on the landscape, that would suggest there might be some gene flow occurring back from tetraploids to diploids. We just don't find them. So the question is wyomingensis derived from basin, mountain, or is it some sort of hybridization between the two of them? And is it a single event, which would be a true subspecies, meaning it occurred once on the landscape and then wyomingensis is spread across the landscape? Or is this subspecies derived from multiple events? And in this case, it wouldn't really be a true subspecies because it doesn't occur from the same origin. I think these questions are fundamental to how we can consider restoring Wyoming habitats and how we organize distributing seeds across the landscape. So real brief on the methods. We took 25 genes that are associated with secondary metabolites. So secondary metabolites are used a lot in plant defense. And we chose these secondary metabolites because we thought we had a chance of finding genes that are actually under selection. If you think about what these defense-related genes are doing, they're usually highly selected on by herbivores. So in total we had over 12,000 base pairs of data. That's each nucleotide that we're counting there. And we took the consensus of all those genes and organized them based on each population or 7 individuals. And we did a phylogenetic analysis, and we also knew the polity of each of those plants as well, or at least 3 in each population. So this is the results of just looking at the diploids across the landscape. So there's over 50 different populations here that we sampled. And you can see that, so each of these nodes represent a population on the landscape. And you can see that all the green show subspecies Bassiana, basically in one group. And then all the basin subspecies in a separate plate here. So that's comes to a bit of relief that we're actually able to separate the diploids. When we add in the tetraploids, things get a little more interesting. If you can pick out the black labels in the tree, you can see we still have separation of our Bassiana and our tridentata subspecies, but our tetraploids are mixed throughout both clades. And I have these labeled as wyomingensis and then Bassiana tetraploid and tridentated tetraploid. You can see that this within wyomingensis, we get populations occur in different clades, even wyomingensis that occurs essentially right in the middle of the Bassiana clade. This population doesn't show any UV fluorescence whatsoever, but yet, based on molecular data, it fits right in. So there's always exceptions to the rule. This is that same tree just shown with a different type of phylogenetic algorithm. It does a better job of showing hybridization. And you can see that basically on the left side you have this tridentata group with your tetraploid, especially your wyomingensis, interspersed throughout it. And again, here's that same Bassiana group with the wyomingensis over here. And then you find these tetraploid tridentata subspecies interspersed between these two groups. So let's see if I have another slide here. It suggests that there's quite a bit of hybridization occurring, probably between tetraploids derived from each group, making up some of these clades, and they seem to be regional too. So all the arrows at the bottom there show populations collected from Arizona and New Mexico. And we have a large, all the ones we collected from there seem to be tetraploid and seem to have some shared characteristics with Bassiana. This is another example where we collected basically three subspecies, all within probably about 10 miles of each other, just going up a elevation gradient. In fact, they all come out being fairly closely related to each other. So another instance where there's probably lots of integration going on between these subspecies at the local level. And then we have probably maybe some larger races of wyomingensis occurring across the landscape, possibly. Our current research is trying to kind of geographically define where these different tetraploid races occur. So what I showed you was kind of the big picture, taking all the genes and essentially a consensus of each population and looking at it that way. The other way you can look at it is take an individual gene and sort them out based on their alleles. So each gene has 2 or 3 or 10 alleles in the genome, depending on what kind of gene it is. And this network shows basically each node shows representation of each diploid subspecies, and the line that connects to another node shows one base pair, one nucleotide difference between these different alleles. So you can see that a lot of our genes do separate out according to diploid subspecies, but you also have some of these larger nodes that have a lot of individuals are mixed. Again, suggesting